Hello everyone, this is Dr. Manpreet Khurana. Today, our topic of discussion is citric acid cycle or Krebs cycle. We also call it tricarboxylic acid cycle. So let's understand what is this cycle all about. We'll try to finish this cycle in 5 to 7 minutes. I've made a small formula or a mnemonic. So we will understand all the substrates, all the products, wherever NADH is produced, wherever FADH is produced, and what are the inhibitors of citric acid cycle and its significance, where it is located, and everything. The formula says OCI, or you can say OCI, OK, SSFM. So this is a small formula which you need to understand. Now, how to learn which substrate is used and whatever products are formed. Let's see that OCI, after OCI, alternatively, NADH is formed. So I'll just highlight these. So after OCI, NADH is formed, then something else, then NADH is formed, then something else. Then here it is FADH. Why FADH? Because if you see here, it is F. So NADH converted into FADH. This is just for learning. Then after one alternate, again NADH in the end. So we have NADH, NADH, FADH, NADH alternately. Now let's fill in the blanks in between. So if you see this one here on the left side is CO2, on the right side is H2O, and in the middle it's ATP. And in the middle it's ATP. Now let's understand what are these substrates and what is the significance. The first thing is NADH. Now all of these products like NADH and FADH, they undergo oxidative phosphorylation. We'll understand oxidative phosphorylation in a different video, but to understand this cycle, let's see that oxidative phosphorylation helps in the production of ATP or energy or adenosine triphosphate. Now from NADH, 2.5 ATP are formed per mole. Okay, so one NADH molecule gives rise to 2.5 ATP. Previously, if you see older editions of the books in the energetics, you will see that one NADH is forming 3 ATP and one FADH is giving rise to 2 ATP. But actually, this was incorrect. So after a long time, what happened is that they actually realized that the exact ATP formed is 2.5. So we don't need to round up the number to 3. We have to use the exact numbers because whatever questions are nowadays coming in the exam, they will use the new energetics. The old energetics are no longer used. Okay, so keep in mind that one NADH gives rise to 2.5 and one FADH gives rise to 1.5 ATP. Okay, this is a very important concept to understand and the newer concept, right? Now, here it is NADH, 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 and one FADH. So right now we are understanding that if one molecule of acetyl coenzyme A is coming into this cycle. Now you might be wondering what is acetyl coenzyme A. Now what happens is the lipids or the fats, the carbohydrates and the proteins. So what happens is they are all metabolized. When they're metabolized, there is a common product or the common product which is going into a common pathway which is known as acetyl coenzyme A. Now this acetyl coenzyme A enters the Krebs cycle. This acetyl coenzyme A is oxidized into various products. So it is oxidized with the use of oxaloacetate into citrate, right? So acetyl coenzyme A is getting oxidized to citrate with the help of oxaloacetate. So let's first understand what is OCI, OK, SSFM means. O for oxaloacetate, C for citrate, I for isocitrate. Then here it is O for oxalosuccinate, K for alpha-ketoglutarate, S for succinyl coenzyme A, S for succinate, F for fumarate, and M for malate. So these are the various intermediate products. Okay. Now here, if you see, this is CO2 and here it is H2O. But here there is one ATP formed here directly at the level of substrate. Now this type of ATP formation is known as substrate level phosphorylation. Substrate level phosphorylation. Now, ATP which is formed at the level of substrate itself is called as substrate level phosphorylation, and ATP formed from NADH or FADH with the help of oxidative phosphorylation. So that is known as oxidative phosphorylation, where one NADH is giving rise to 2.5 ATP and one FADH is giving rise to 1.5 ATP according to the latest concept, right? Now, if you see the cycle, now let's see what all enzymes are involved. Okay, so enzymes, learning the enzymes is very, very easy. Why? Because we know wherever NADH is formed, wherever ATP is formed, and wherever no substrate is formed, no product or no intermediate product is formed. Now, whenever there is NADH or FADH formation, the name of the enzyme is usually the name of the enzyme is dehydrogenase, because this is a type of dehydrogenation reaction. Whenever there is formation of ATP, the name of the enzyme is kinase. That it ends in kinase. Now, with this formula, we can even understand the enzymes. Now, let's see first enzyme. Oxaloacetate with the help, like acetyl coenzyme A is oxidized by oxaloacetate into citrate. So here something is formed. What is formed? Citrate is formed. So the name of the enzyme is citrate synthase. Synthase means synthesis. Then citrate is converted into cis-aconitate, right? Now, this reaction is important because this reaction is inhibited by fluoroacetate, right? So if you see here, I have marked it as negative sign. So fluoroacetate inhibits this reaction where citrate is converted into cis-aconitate and the enzyme is aconitase. So you have to learn this enzyme. Now, what is happening is 6-aconitate is converted into isocitrate. Now, again, the same enzyme aconitase is taking into consideration. So the same enzyme is, following this reaction. Now, from isocitrate, we are getting oxalosuccinate. Now, this is the first NADH where it is formed. And as I told you, whenever there is NADH, the name of the enzyme is dehydrogenase. So the substrate is isocitrate. So the name of the enzyme is isocitrate dehydrogenase. Okay. Now, after this oxalosuccinate, there is formation of alpha-ketoglutarate, right? So alpha-ketoglutarate, there is CO2 is released here. Okay, one CO2 is taken out. Now, here also the same enzyme is occurring, that is isocitrate dehydrogenase, right? From alpha-ketoglutarate, there is formation of succinyl coenzyme. Again, NADH is formed. So the name of the enzyme is alpha-ketoglutarate dehydrogenase, right? So name of the substrate NADH है तो dehydrogenase, FADH है तो dehydrogenase, ATP है तो kinase. Okay, so here it is alpha-ketoglutarate dehydrogenase. Now from succinyl coenzyme there is formation of succinate. Here it is known as succinate thiokinase. Why kinase? Because यहाँ पर ATP is formed, right? So succinate thiokinase. Now from succinate there is formation of fumarate, right? Now again, if you see in this FADH is formed, the name of the enzyme will be dehydrogenase. So the name of the enzyme is succinate dehydrogenase. Okay. Now from fumarate there is formation of malate. The name of the enzyme is fumarase, and from malate there is formation of oxaloacetate. Now if you see in the end also there is formation of NADH. So the name of the enzyme will be dehydrogenase. So name of the substrate with dehydrogenase, that is malate dehydrogenase. Okay, now let's review the cycle again because there are a lot of things which we have talked in a very short time. Let me just give you the formula. It's OCI OKSSFM. O stands for oxaloacetate, C for citrate, I for isocitrate, O for oxalosuccinate, K for alpha-ketoglutarate, succinyl coenzyme A, succinate, fumarate, and malate. After OCI, alternatively NADH or FADH is formed. FADH कहाँ पे बनेगा? जहां पर F आएगा, that is fumarate. So FADH, NADH, NADH, NADH. Okay. Now, in between these alternate NADH, there is formation of left side CO2, right side H2O, and in between ATP. Whenever there is NADH, the name of the enzyme is dehydrogenase. Whenever there is ATP, the name of the enzyme is kinase. For example, if I ask you a question, what is the name of the enzyme when isocitrate is getting converted into oxalosuccinate? The name of the enzyme will be isocitrate dehydrogenase. Here, alpha-ketoglutarate is converted into succinyl coenzyme A. The name of the enzyme is alpha-ketoglutarate dehydrogenase. Here, succinate is converting into fumarate. The name of the enzyme is succinate dehydrogenase. Again, malate is again converting into oxaloacetate. The name of the enzyme is malate dehydrogenase. ATP is formed here. The name of the enzyme is succinate thiokinase. So ATP means kinase, right? So now we can remember all the enzymes just by knowing this formula, where NADH is formed, where FADH is formed, and where ATP is formed. Thank you and have a nice day. Before that, let's see the energetics of citric acid cycle. Now we are learning citric acid cycle. Glycolysis will cover in a different video, right? If you see wherever there is NADH, we are taking two molecules of NADH. Why? Because there are two molecules of acetyl coenzyme A which are generated. So from one acetyl coenzyme, there is one NADH. From two, there will be two NADH. So there will be two NADH when isocitrate dehydrogenase is there. This pyruvate dehydrogenase complex is formed, is used, right? Is utilized, sorry, when pyruvate is converted into acetyl coenzyme A. So what is happening is the pyruvate gets converted into acetyl coenzyme A, and acetyl coenzyme A is helping in the citric acid cycle. It is entering the citric acid cycle, right? So there is formation of 2 NADH here as well. So 2 NADH here, then there is substrate level phosphorylation, which I have told you at the time of succinate thiokinase, where there is ATP is formed, then FADH2 and NADH, right? So one NADH is giving rise to 2.5, 2 NADH will give rise to 5. So here also 5 ATP, 5 ATP, 2 ATP, 3 ATP, and 5 ATP, because one NADH gives 1.5 ATP, 2 will give 3. One NADH will give 2.5. So 2.5 multiplied by 2 is 5. So they will give 5, 10, 15, 20, 23, and 2, 25. So net ATP formation is 25 from citric acid cycle. Okay. Thank you and have a nice day. And don't forget to subscribe and like my video and subscribe my channel. Thank you.